Good morning, students. Welcome to lecture 5. Hope you are well and good at home, like staying at home and taking care of yourselves. So I got a question from one of the students asking me that whether the exam will happen on April 13th. Yes, it is going to happen on April 13th. I'll post the details. Now, about the questions. Well, I cannot give you short answer questions because there is the TAs are not available physically to grade those questions. So we will depend on multiple choice or fill in the blanks or matching type of questions. Although I haven't really decided how the questions will be distributed, but mostly it will be like single point questions or double point questions, like 1 point each or 2 point each, something like that. Uh, if you know the material, you should be able to understand, answer the questions. There is no worry in terms of the difficulty because I know that this is a big shift this time, so the questions will be relatively simpler this time. So, but then you have to know the material, I'm telling you again and again. So before beginning to lecture 5, we kind of stopped last time, lecture 4, with the um, two different models for intra-Golgi trafficking. And we discussed about the vesicular transport model and the cisternal maturation model. I'm not going to ask you to kind of explain those two models, but you know that just cisternal maturation model has been accepted worldwide. And later on, we'll see that the coat proteins are extremely important. So do know about the coat proteins that we have, either COP1 or COP2 or AP2 clathrine coats that we are going to discuss later on. So coming back to the lecture 5, this is all about the vesicular traffic, secretion, and endocytosis. So we will be discussing about proteins which are either secreted from the transgolgi network onwards towards the cell surface, or the proteins which are taken from outside the cell surface or the plasma membrane inside the cell to be endocytosed. Endocytosed means basically most of the time they are basically taken inside the cell for degradation. So it's the later stage of the secretory pathway because we have seen the proteins getting transported from the ER to the transgolgi network through the cisternal maturation model. In the later stages, we need to focus on few important points, that the transport happens via vesicles targeted for different destinations. Now this point is important, targeted for different destinations based on the coat proteins that gets formed. We'll discuss that in a bit. And the tags, different tags are associated with proteins which are specific to certain transport cycles to be taken to specific destination. For example, if it is a lysosomal protein, that means a protein which is destined to go to lysosome eventually. So those kind of proteins will get a tag that is called M6P tag, mannose 6-phosphate tag, and those tagged proteins will go inside the coated vesicle, and then that coated vesicle will specifically take that enzyme to that lysosome. Okay, so similarly in this section we are going to talk about two different tags. One is for the LDL receptors and one is for the lysosomal enzyme receptors. And as we know that in case of secretory proteins from the ER through the secretory pathway, the proteins which are destined for the plasma surface or plasma membrane surface or outside, there are two kinds of secretion that can happen. One is called regulated, so that whenever there is extracellular signal from outside the cell that the cell requires more protein, then only those pathways gets activated. Otherwise, the proteins gets uh, stuck in the vesicles and stay inside the cell. Okay. For the constitutively secreted proteins, the proteins actually keeps on getting secreted outside because there is no control over it. It's just a continuous process. And also, interesting fact is that some proteins are processed into mature form after leaving the trans-Golgi network. Because so far what we have seen is that most of the proteins gets folded properly inside the ER, especially for the secretory proteins. And over the period of time, like from ER to cis and cis to medial, medial to trans and trans to trans-Golgi network, every compartment gets its identity because of the presence of specific enzymes which help in the maturation of side chains of the oligosaccharide side chains associated with the proteins. Basically, it basically helps to mature the whole protein. But in some instances, the proteins gets matured once they leave the trans-Golgi network and then get into the vesicles to be secreted outside. Now, these quotes are very important. When I say quotes, so remember that these are extremely important because these quotes define where they are supposed to go. That means these vesicles containing particular proteins, obviously those proteins will have their specific tags where they are supposed to go. So for example, if we start from here, so this is the trans-Golgi network, this is the trans-Golgi. So it is basically backtracking. So this one uses the COP1 code. And we have discussed that COP1 code is for retrograde transport. That means either from cis-Golgi to the ER for the missorted proteins. Also during the cisternal maturation, it is from trans-Golgi network to trans and then trans to medial, medial to cis to transport the essential enzymes so that it can change the next compartment's nature, okay, and also helps in the maturation process of the protein. So in this case, we have COP1 coated vesicles, step number one. The step number two is basically AP complex. It may have Clathrin coat as well that we haven't discussed yet. We are going to how the Clathrin coat looks like. So if some vesicles gets Clathrin coated, then they are supposed to go to lysosome. Either AP or Clathrin coat or both, they're supposed to go to lysosome. The third one is Clathrin coat plus AP2 vesicles. When I say AP2, that means there are different kinds of AP. AP means adapter proteins. So those adapter proteins are basically three different types, AP1, AP2, AP3. In this case, the step number three, the red color, the Clathrin coated plus AP2 vesicles. That means both sets of proteins are present. They basically transport the lysosomal enzymes. That means the enzymes that are destined to lysosome, and then they make these vesicles bud off, and then they mix, go and fuse with the late endosome. Now, you may ask that if the proteins or the enzymes are supposed to go to the lysosome, what are they going to, why are they going to late endosome? Well, sometimes the lysosomal enzymes directly go to the lysosome, and sometimes the lysosomal enzymes go via late endosome. So the step that is missing here is from late endosome getting fused to lysosome. Now the fourth and fifth step that we already know from earlier, the fourth one is the constitutive secretory vesicles. The coat that forms around is not known in detail, so we are not going to talk about that. And the fifth step is for the regulatory proteins. That means the proteins which are regulated in terms of secretion as per need basis. So what kind of proteins gets transported or secreted outside? So for example, if it is a constitutive secretion, the cargo proteins inside that are to be transported outside the plasma membrane can contain ECM proteins, that is extracellular matrix proteins, the blood proteins, the immunoglobulins, etc. And in case of regulated secretion, the proteins can be the digestive enzymes and peptide hormones. Now, it all makes sense, right? Because we cannot have secretion of digestive enzyme throughout. I mean, without any control, we cannot have, because in that case, we'll get acid reflux 24 hours a day, except the times when we are eating something and the stomach is trying to digest something. Other than that, whenever we are not eating, that time also, otherwise, the digestive enzymes will keep on getting produced and keep on getting secreted, and will eventually get acid refluxes, which is bad for the body. Obviously, that is why it does not happen. So the cell has made it very particular that whenever certain proteins are needed, only then those will be released outside. The second type is peptide hormones. We know that a hormone level should always be maintained in the blood. If it gets more, then it's a hyper condition. If it is lower than the normal amount, then it's the hypo condition. And there are feedback mechanisms and all that you have studied in ANP2 and ANP1. So not ANP1, ANP2. So those are, so both in both the cases for the constitutively secreted and regulatory pathway, the actual code proteins are not known in detail. So we are not going to discuss the code proteins here, but the major ones, COP1, that's the retrograde. Do you know the anterograde? What kind of code proteins are used? It is the COP2 coded vesicles. And then we have AP or clathrin AP. This is clathrin plus AP2. Okay, going to the next slide. This is how a clathrin coat looks like. So this is basically a cryo-EM reconstruction. That means the whole structure has been freezed, and then freeze structure basic it's freeze, not free. Freezed or FREEZED. Freezed structure is being looked upon through this electron microscope, and this is how it looks like. So this whole clathrin coat assembly, now unlike the COP2 coat assembly, we are not going to discuss how the coat assembly happens here. We also didn't discuss how the coat assembly happens in COP1 also. So this is just the structure. So there are heavy chains, three big heavy chains with the globular domain at the end, which is the binding site for assembly particles. And there are smaller light chains. Now, these are 180,000 molecular weight, and these are 40,000 molecular weight. So what do you mean by molecular weight? Because we always know that protein weights are expressed in daltons or kilodaltons. So just for your reference, just know that one molecular weight is equivalent to 1 gram per mole, which is equivalent to 1 mu or 1 Dalton. So 180,000 molecular weight is basically 180,000 Dalton or 180 kilodalton. Similarly, this is 40 kilodalton. That's the size of the protein. And these intrinsic curvatures are due to the heavy chains. So these are basically composed of 36 triscallions. The triscallions means three-legged from the Greek term. And if you remember the uncoating, so if the coat formation happens, then obviously for the target membrane fusion, we have to have a coat disassembly as well. So if you remember the COP2 codes specifically, that SAR1 was the GTPase which did the hydrolysis, GTP hydrolysis, and SAR1 GTP hydrolysis provided the energy to basically depolymerize the coat once all the cargo proteins were assembled and recruited inside the vesicle before the fusion. In this case, do you know what is the triggering factor? Because if you see the chart that is there earlier in earlier lecture, you can see that this is ARF in this particular case. This ARF GTPase basically helps in the hydrolysis, and that whole process is being mediated by the cytosolic HSP70 activated by oxalin, which is a co-chaperone. So in this particular case, uh, the ARF GTP hydrolysis is being mediated by cytosolic HSP70, which is also a chaperone. Do you remember what mediated the SAR1 or SAR1 hydrolysis in case of GTP hydrolysis in case of COP2 vesicles? It was SEC24, SEC24 protein, okay, which was also being recruited by SAR1. So from this, not asking you like how many units it is composed of or something, or the molecular weights, but you do need to know some facts, which is this intrinsic curvature is due to the heavy chains. There are three sets of heavy and light chains present. Also, the coating is done by ARF, and the or recruitment of coat proteins are done by ARF, and hydrolysis of ARF GTP is done by or mediated by cytosolic HSP70. Now, as we mentioned that we are not going to go into details how exactly the coat formation happens in case of Clathrin AP coat, but we do need to know how they are pinched off. That means the vesicle forms, obviously, and the soluble cargo proteins are recruited, and the membrane proteins are recruited on the surface of the vesicle, coated vesicle, and then the vesicles need to get pinched off from the surface, right? And only then the actual depolymerization process can start, and only then the vesicle can fuse with the target membrane to deliver the cargo proteins or enzymes. So in this case, there is a particular molecule here, a particular protein that is called dynamine. Okay, so dynamine mediated pinching off of clathrin coated vesicles. Now, few things are important here. First of all, whenever we are saying clathrin AP2 coat, so for example, in the previous slide, we saw clathrin assembly, but we did not see the AP coat. Well, that is because the AP coat is basically the inside coat. This blue structure here around the vesicle, that is the AP complex. And the AP complex or the adapter protein complex is the inner coat and is three different types, AP1, AP2, AP3, each with 4 subunits each. Okay, so the AP coat is inside. So from by looking from outside, we can only see this clathrin coats. And you can see these triscalene structures, the red structure all around the circumference, that's the fibrous clathrin coat. And these coat proteins basically helps in the recruitment of these soluble cargo proteins and the integral receptor proteins. Now, if you remember the receptor proteins, there was a specific receptor protein that was basically receptor signal, or that was diacidic arginine signals, right? Excuse me. So this second point is this pinching off by the help of these dynamine molecules. These dynamine molecules are required for pinching off of these clathrin coated vesicles, not the COP2 or COP1, but specifically the clathrin coated vesicles. These dynamine molecules, they are several in number. This is just one that is showing. There are several in number. So all of those molecules gets polymerized around this neck portion, right? And then there is a GTP hydrolysis associated with the dynamine that happens, and that provides enough energy to actually pinch this bud off from this surface, okay, and make it an independent coated vesicle. Now, the sorting signal that's being used by AP1 Clathrin coat is this. I'm not going to ask you to remember this sequence, but just know that this is the sorting signal recruited. When I say that sorting signal, for example, this is an integral receptor protein, right? And then this integral receptor protein recruits a soluble cargo protein to be transported to the destination. So this has the sorting signal, specific sorting signal, which recognizes and recruits specific cargo protein, soluble cargo protein. And for the sorting signal for AP2, remember this second AP type is also called GGA adapter protein, either AP2 or GGA. So for the AP2 Clathrin coat or the GGA Clathrin coat, this is the sorting signal sequence. Okay. There is one more, that is AP3, that we did not discuss in terms of sequence, but AP3 is very important because AP3 is required for the delivery of proteins to the melanosomes as well as the megakaryocytes. Now, excuse me, what is melanosome? Melanosome are basically uh, an area of vesicle which contains the black pigment melanin in skin cells, okay, which gives the coloration of the skin, lighter skin, darker skin. So we all have heard about melanins. So melanosomes are the vesicles that basically has those melanins accumulated inside. So AP3 in particular helps in the delivery of proteins to melanosomes, uh, which contains the black pigment melanin, and to the platelet storage vesicle in megakaryocytes. That is, the megakaryocytes are the locations where the platelets are accumulated. Okay, so large cells, these are large cells that fragment into dozen of platelets. For example, if there is a mice with mutations in either of two subunits, two different, remember each of these AP complexes are made with four subunits. So if there is a mutation in mice where there are the two of the subunits in AP3 are mutated, in that case, they not only will have the abnormal skin pigmentation because the melanins or the proteins cannot be transported to the melanosomes properly, but also will exhibit the bleeding disorder. Okay, so that's why AP3 is very important because they help in the delivery of proteins to melanosomes as well as megakaryocytes. Now, going to the next slide, how exactly this happens, or in general, a broader overview. Okay, so let's assume for a second that this is actually a nerve terminal. So here we can see that vesicle gets formed, but it's not pinched off. Rather, the neck has been elongated. So what does that mean? That means in this particular case, to understand how exactly the dynamine works, what scientists did, because we know the other fact is that GTP hydrolysis is necessary. So what they did is that they used an abnormal excessive amount of GTP gamma S. GTP gamma S is the non-hydrolyzable derivative of GTP. That means a GTPase which cannot be hydrolyzed. So there is no question of energy production. And if there is no energy production, what will happen? The dynamines will keep on accumulating here. Excuse me. The dynamines will keep on accumulating here, and it will just extend the whole neck portion, but it won't be able to pinch off this vesicle from the surface membrane because there is no energy. Okay. Now this also can be done in another way. So for example, if you mutate the GTPase, then also in that case the GTP won't be able to hydrolyze, so there won't be any energy, and the dynamines will keep on getting accumulated. And once we have regular GTPases, in that case we can have the proper hydrolysis of GTP and pinching of this vesicle. Okay. Now, I was talking about the receptors. So two of the receptors are very important that we are going to study in details. One is the M6P receptors. Those M6P receptors are tagged to the proteins or the enzymes to be transported to the lysosome. Okay. And the other one is LDL receptor that we'll discuss later. So let's talk about this formation of mannose 6-phosphate M6P residues that target soluble enzymes to lysosomes. So as the name suggests, it's mannose 6-phosphate. That means I'm pretty sure that in your biochemistry 1 and 2, you have studied the structures of glucose, fructose, mannose, etc., ribose, deoxyribose, all those structures. So the sixth position on the mannose residue, there is a phosphate group associated. That is why it's called mannose 6-phosphate. So what happens in this particular case, the M6P assembly to the targeted proteins targeted to the lysosome has to happen. That gives the identity to that protein basically. So how is the whole process that occurs? So the first thing is that we have in the cis-Golgi glucnac phosphotransferase. If you remember, we have already discussed about this. The glucnac phosphotransferase, what it does, as the name suggests, glucnac as well as phospho transfer. So there is this UDP associated, UDP glucnac associated, that is uridine diphosphate and glucnac residue, that red block. And then we have this protein, which is supposedly lysosomal targeted protein, lysosomal enzyme, let's call it. And along with this oligosaccharide side chain with all these blue circular structures, those are mannose residues, right? So the glucnac phosphotransferase, what it does, it basically transfers this particular phosphate residue along with this glucnac residue. There is this glucnac plus phosphate residue to the sixth position of one of the mannose residues. And how many mannose residues are there? There are 1, 2, 3, 4, 5, 6, 7, 8 residues now. Okay. Excuse me. 8, yeah, approximately 8 residues. So once this glucnac and the phosphate together gets transferred with the help of glucnac phosphotransferase, then there is another enzyme that is called phosphodiesterase. What it does, it basically cleaves this glucnac residue from the glucnac phosphate residue so that this mannose residue at its sixth position can only have the phosphate residue attached. And that is what is called M6P tagging or M6P receptor. So basically two major enzymes required for the addition of M6P residue to lysosomal enzymes. The first one in the series is glucnac phosphotransferase, which transfers a glucnac phosphate residue. And the second one is the phosphodiesterase, which cleaves the glucnac residue and keep the phosphate residue attached to the sixth position of one of the mannose residues. Now, these M6P residues are being recognized by the M6P receptor. Now, you can ask, where is the M6P receptor? Well, if you remember, this is the soluble protein, right? And the soluble protein with a tag. So soluble protein, soluble cargo, and also this will be a cargo protein because it has to be transported across. So the soluble cargo proteins are recruited by the receptor proteins, membrane receptor proteins. So the membrane receptor proteins which identifies these M6P receptors are called M6P receptor proteins. Okay. Okay. Uh, this diagram, it looks somewhat complex, but it's not really. Uh, this diagram properly explains about the M6P and the M6P receptor. Now we are talking about the secretory proteins which starts from the trans-Golgi network, obviously. So it can either go, since it's secretory, it can either go to the plasma membrane and get secreted outside, right? The M6P receptors are these receptors, okay? These orange color ones on the membrane of trans-Golgi network. So when the vesicle buds off, the few of these receptors also get inside these vesicles and they get transported, but they do not get released outside since these are membrane proteins. So they remain stuck in this plasma membrane M6P. Okay. So let's talk about the first step itself. So we know that in the cis Golgi there are two sets of enzymes which helps in the maturation. That means M6P tagging of the protein to be transported to lysosome. And then once the protein reaches to trans-Golgi network, bud formation happens along with clathrin and AP coat. And then from here, this becomes independent vesicle after the pinching off of the vesicle. Remember what is the protein that basically does that? It is dynamine. Okay. Now from here, the depolymerization of the coat protein happens, and then now we have this vesicle, uncoated vesicle with obviously V smears because exposed on the surface, and also these M6P receptors which recruits the M6P molecule attached to the cargo protein. Now from here it can go to the late endosome. And in the late endosome, what happens? This M6P receptor releases this M6P tagged protein. Why? Because of the low pH. Okay. And then from here, since this M6P receptor itself is a membrane protein, another vesicle forms, coated vesicle with AP, and then it goes back or recycled back and gets fused to trans-Golgi network to actually make this cycle happen again. Okay, so that is how this M6P receptors gets recycled back. And here, since due to the low pH, we have release of M6P tagged protein. Now the M6P, that is the phosphate at the sixth position, also gets released due to the enzymes that is present here. And when they get inside the lysosome, obviously they go to the lysosome to be degraded, right? Now, remember we had this step where the M6P receptor actually went to here, or rather from here to here on the plasma membrane. Now, there are certain proteins which needs to be degraded, but which are outside the cell. So we need to bring those back, right? And that is being done by the formation of uh, receptor mediated endocytic vesicles. So the receptor mediated, in this case, the receptors are M6P receptors, which recognizes those proteins which are to be delivered to lysosome to get degraded. And they also form this clathrin coated bit along with AP, and then independent vesicle formation, coated vesicle, depolymerization, and the same process. Late endosome delivery, low pH, release of receptor and the phosphate, and then eventually getting fused with the lysosome to be degraded. That is the whole process of trafficking of soluble lysosomal enzymes from trans-Golgi network as well as from the cell surface to lysosome through receptor mediated endocytosis. Okay. Excuse me. Now there is something called eye cell disease and M6P receptor. So how M6P receptor is involved with eye cell disease? Okay. So in these kind of cells, the cells actually lack the N-acetylglucosamine phosphotransferase, the first of the two enzymes which is required to tag the M6P receptor to the cargo protein. So the first enzyme is missing. That means the lysosomal enzymes lack M6P targeting signals, right? Because if the M6P cannot be attached to the cargo protein, obviously it won't have the M6P signal. That means unless and until the protein gets tagged with the M6P and gets recognized by M6P receptor and gets transferred to the lysosome, the proteins won't degrade. So if it doesn't have in the first place M6P tag, in that case the proteins which are supposed to be degraded keeps on accumulating inside the cell. Okay. So the undigested lysosomal contents was continued accumulation forms inclusions which may eventually kill the cell, and undegraded debris cause developmental and physiological and neurological abnormalities. Okay, so in that case, in vitro cure is if we can add M6P enzyme, that is N-acetylglucosamine phosphotransferase, to the cell, we can cure this. So in this case, for the eye cell disease, in case of the fibroblasts, we add the M6P externally, and those gets internalized by endocytosis pathway and function on arrival in lysosomes. Okay, so there is that. So several questions can be asked from this particular figure or picture. So for example, M6P gets recognized by M6P receptor where? Okay, so there are actually two different pathways. One obviously is the secretory pathway, and the other one is to the soluble lysosomal enzymes to the lysosome. Okay, so for the soluble lysosomal enzymes, M6P receptor is on the trans-Golgi network. Okay, this is the trans-Golgi network because from here the bud will form, and it forms the clathrin AP1 coat. Okay, this is the clathrin AP1 coat. For the secretory one, that means the secretory lysosomal enzymes, M6P receptor is at the plasma membrane. Okay, that means the secretory proteins which are outside, they are supposed to be taken inside. So here the clathrin and AP2, the coat that gets formed has AP2. Here it is clathrin AP1. Here it is clathrin AP2 coat is at plasma. Here is at trans-Golgi network. Here is at plasma membrane. Okay. Now I think we'll stop this section here and we'll resume the section in a bit. Okay. Hello, student. Uh, Welcome back to the lecture 5, the second session. So we stopped here basically for the M6P receptors. We stopped here where we saw that M6P receptors are basically present for the lysosomal enzymes. They are present on the trans-Golgi network here, and for the proteins which are to be receptor mediated endocytosed and taken back to the lysosome, the receptors are present on the plasma membrane. Okay, and then we also discussed about eye cell disease, that if the M6P receptor, sorry, the M6P tag doesn't actually form due to the lacking of glucanac transferase, phosphotransferase. So those proteins gets accumulated and causes diseases, especially developmental diseases. Okay. So let's come to the next section, which is the receptor-mediated endocytosis for the LDL particles. Okay, so in this case, Obviously, since it's clathrin coated vesicles and the molecules are coming from outside of the cell to the inside of the cell and basically taken to the lysosome. So the coat formation happens with the help of clathrin and AP2. Okay. So here we are talking about the delivery of the ligand, that is the LDL particle, and into the late endosome, the acidic environment, the dissociation happens for the most receptor ligand complex. In this case, uh, for the LDL receptor, the dissociation happens, but for another example where we'll talk about the iron, in that case, the receptor and ligand dissociation does not happen, but that's later on. So pH is very important in terms of dissociation of receptor from the ligand. Okay. So talking about the LDL itself, what is LDL? So it is called low density lipoproteins, that is LDL. LDL is called bad cholesterol because it takes cholesterol to your arteries. So whenever there is, you have heard that, some people have deposition of cholesterol in their arteries and blockage of arteries causing stroke or heart related diseases, right? It also increases the blood pressure. So what happens is that somehow if these LDL particles are not digested in the lysosome, these LDL particles keeps on accumulating inside the vessels, okay, outside the cell. In that case, a layer of LDL particles forms in the inner surface of artery, causing deposition or the atherosclerosis, basically. So, if we talk about this one particular LDL particle, this is one single particle, by the way, okay, we can see that this is a phospholipid monolayer, which is unique because we usually come across in biology, we usually come across bilayer, phospholipid bilayer. But in this case, this is phospholipid monolayer. Okay. And on the surface, we have unesterified cholesterol, but inside it is packed with cholesterol ester or cholesterol, basically. Okay. This is the apolar surface, this is the polar surface, and that is why it is called amphipathic cell. That means both polar and apolar surface are present. Okay. So there is that. And the lipoproteins are used to facilitate mass transfer between cells, thousands of lipid molecules, right? And they are packaged into water-soluble macromolecular carriers. So this is water-soluble macromolecular carriers. This is called lipoprotein, okay? Now, coming to the next slide, we can see, just like previously we have seen the internalization, basically when we discussed about the snipping of the bud or pinching off the bud by Dynamine, it is similar kind of mechanism because since it is involved this clathrin and AP2 coat, and we know that Dynamine is involved wherever there is clathrin and AP2 coat is present. So this is the outside of cell surface, this is the plasma membrane, this is receptor mediated endocytosis. That means the vesicle gets invaginated, forms inside of the plasma membrane, and we have tagged the LDL particles with the electron dense labels, specifically iodine, radioactive iodine, and it shows that initial stages of receptor mediated endocytosis or RME reveals by electron microscopy. Okay. So for this one, the 4 degrees centigrade is the restrictive temperature. So what do you mean by restrictive temperature? Well, these are the cultured fibroblast cells. These cells are basically treated with these electron dense labels, LDL particles. Okay. At 4 degrees centigrade, the LDLs binds to the LDL receptors and the receptor mediated endocytosis process begins right? But it gets inhibited. That means this vesicle formation or the pinching of the vesicle doesn't happen. Initial invagination happens. All the LDL particles, tagged LDL particles, just accumulate around this vesicle or the invaginated groove. But whenever we change the temperature to 32 degrees centigrade, which is the permissive temperature, then these LDL particles or the tagged LDL particles gets inside these vesicle. And then here, obviously, the depolymerization happens. We cannot see really, but there has to be a coat, which is Clathrin AP2 coat. Then the depolymerization of the coat happens, and then this particular vesicle gets infused with the endosome. So the LDL receptor complexes are delivered to the endosome. It takes about 6 minutes from here to here. 6 minutes after the internalization begins. So this is the internalization. Okay. Uh, in this case, so if we just plot the earlier picture into a graph, what are we going to see is that this is the binding. So this is a pulse because we are using the radio labeled radioactive iodine 125 isotope. So we are doing a pulse. So this is the whole time is about 2 hours. So we are doing a pulse for brief exposure of LDL, like radio labeled LDL particles. And then at 4 degrees centigrade, all the particles are basically at the surface of the plasma membrane from outside, or the invaginated portion of the plasma membrane, but on the outside surface. Okay. But as soon as we change the temperature to 32 degree, which is the permissive temperature, immediately the binding starts. Binding means binding to the outer surface of the invaginated plasma membrane. Okay. So the binding happens, and binding slows down a little bit. Why? Because as the binding happens, at the same time, the internalization, that is the vesicle budding off inside the cell, that keeps on happening, which also have all those tagged LDL particles. So let's say we have 100 particles in the beginning. Just assume there are 100 particles in the beginning. So at this point, all the 100 particles are moving around freely here outside the surface. As we change the temperature to the permissive temperature, immediately internalization begins. And then as the internalization begins, at the same time, so we can see that increase in the internalization. So not all the particles gets into the vesicle together at the same time, but the buds keeps on forming. So that is why we are seeing that internalization begins, and then when all the molecules eventually gets internalized, then obviously there are no more molecules outside the cell surface. So this becomes straight line, and then this also slows down because there is no more molecule to internalize. So about somewhere after 12 minutes or 15 minutes, about this lag phase, once majority of the molecules, or at least several of the molecules gets internalized, then those vesicles keeps on getting fused with the late endosome, okay, and go for the degradation. And as the internalization increases, the amount of particle degradation also increases. Okay, so the event is binding of LDL, internalization, and then degradation, right? So this graph basically shows the earlier picture that we saw, right? And there is a lag phase because it takes some time to bind and then internalize. That is why this lag phase is there. Going to the next slide, we have the actual process of how the LDL receptor LDL particle works. Okay, so we have this LDL particle, and I completely missed telling you about this apolipoprotein here. So this is a single molecule apolipoprotein or ApoB. Sometimes it's called ApoB 100. It is a molecule around the circumference of this LDL particle. And this is very important because here we are going to see that that particular ApoB protein is being connected by the LDL receptor. So this whole structure is called LDL receptor, just like M6P receptor on the plasma membrane captures the proteins destined to go to the lysosome. These LDL receptors captures the LDL particles specifically, which are destined to go to the lysosome to get degraded. So this LDL receptor has two different arms. This pink portion is called extracellular domain, which binds the LDL particle, but particularly in this ApoB band area on the LDL particle. That is why ApoB is so important. Okay, and then we have this small green portion which is embedded in the walls or the coat itself, or basically it's being recruited by the coat, and that is called cytosolic tail. And this signal that gets recruited, remember sorting signal we discussed earlier? So in this particular case for the LDL receptor, the sorting signal that gets recruited by these AP2 complexes is called NPXY sorting signal. Okay, so this sorting signal is NPXY, which inside forms a cytosolic tail and then extracellular domain. This extracellular domain binds to the ApoB protein and recruits this LDL particle, right? So the coat formation basically helps in the recruitment of particular cargo protein. In this case, it is the LDL particle, the whole particle itself. And then the invagination happens, and what happens next? Obviously, since it's the clathrin AP2 coat, so we have this pinching off with the help of dynamine. Okay, and then it becomes independent coated vesicle, which obviously has to get uncoated or depolymerized so that it can go and fuse with the late endosome. Late endosome is also called curl compartments. So here at the surface of the plasma membrane, we have pH 7, which is neutral pH. But here, the pH in the late endosome is very low. It's about pH 5 or acidic pH. So this acidic pH helps the release of this ligand, which is the LDL particle, from its LDL receptor. Okay. Now this LDL particle goes to the lysosome. Basically, the late endosomes fuses to the lysosome, and here the LDL particle gets digested. So the enzymes present are protease, which hydrolyze the surface ApoB, that particular band here, and the cholesterol esterases hydrolyze the cholesterol esters. That is how, and then the nutrients gets absorbed by the cells. That is how the LDL particle or the bad cholesterol gets removed from the blood circulation. Okay. Now what happens to the receptor? The receptor obviously goes back to this surface to get recycled. Okay, and at neutral pH, the ligand binding arm is again free to bind another LDL particle here. Okay, this whole recycling process takes about 10 to 20 minutes, total 20 hours of life. So every 10 to 20 minutes, this receptor captures, takes them back to the late endosome, and then the receptor goes back or recycled back to the surface. In case of earlier, we studied M6P receptor, each cycle is about 5 to 10 minutes. Okay, so it takes a little bit more time to recycle the receptors back to the surface. Now, if we actually see that why is this structure or the receptor, LDL receptor, binds to the ligand or this LDL particle at neutral pH, and why it releases at low pH. So this is at the cell surface where the pH is 7 or neutral pH. We have this NPXY signal which gets recruited by the AP2 coat protein. Okay. So here is the beta propeller domain, and if you, okay, so here is the beta propeller domain, which is basically histidine rich. And then we have this ligand binding arm, the pink portion. It contains the cysteine rich repeats. So if we take just this structure and see its crystallographic structure here, we can see this is the beta propeller domain, right? And here this is histidine rich, so this is positively charged. And these are 7 cysteine rich repeats, R1 to R7. And in case we are seeing that when it is locked state, that is in the endosome, this R4 and R5, the major ones which are actually connected to the beta propeller domain, because this is positively charged, this is negatively charged. Okay. Now, here we got it that cell surface neutral pH, it binds to the, this arm is, ligand binding arm is free, so it can basically capture the ApoB on the surface of LDL particle. When it goes to the endosome, the pH becomes less, that is acidic pH about 5. Okay. So here what happens, that surface of the beta propeller domain becomes positively charged because it has histidine, and then it basically binds this to the ligand binding arm. So when it binds to the ligand binding arm, the ligand binding arm basically releases this ApoB. Okay. And that is why the LDL particle gets released, right? So there is a disease called familial hypercholesterolemia. This disease, hypercholesterolemia, it is self-explanatory. That means familial means found in the family tree. Hypercholesterolemia means hypercholesterol, which is more cholesterol. Okay. So in this particular case, what happens is that one single amino acid is mutated in the NPXY sorting signal here. Because of one single mutation in one of the amino acids, what happens is that this whole structure, whole LDL receptor is unable to bind to the LDL particle or the ApoB portion properly. And if it cannot bind, obviously the LDL particle cannot be internalized to get degraded in the lysosome. In that case, more and more of the LDL particles will circulate outside the cell, that means in the bloodstream, and eventually get deposited on the surface of the arteries. So that causes this particular mutation on the NPXY sorting signal causes this familial hypercholesterolemia. Okay. And also cause cardiovascular diseases because of this mutation. It causes too little LDL uptake or clearance into the cells. Okay. Now, going to the next one, which is very unique. Why? Because so far we have seen that the receptors are getting recycled back, along with the ligands are getting released in the late endosome compartment where the pH is less. In this particular case, this is unique because this receptor mediated endocytic pathway delivers the irons, okay, irons without dissociation of this receptor with the ligand. The whole setup, the whole receptor ligand complex gets recycled back to the surface of the plasma membrane. Okay, so let's see here. So this is ferrotransferrin, that is ferrotransferrin molecule containing ferric ions, Fe3 plus ions. So the dimer of it binds to the transferrin, excuse me, transferrin receptor on the cell surface. And we know that outside the cell surface, the pH is neutral, that is pH 7. So this ferric ion attached ferrotransferrin binds the transferrin receptor, which is on the surface. Now, once it tightly binds, obviously it gets internalized. And as we know that for receptor mediated endocytosis, it is usually clathrin plus AP2 coat. And since it is clathrin plus AP2 coat, the pinching of the vesicles will be done by dynamine activity. Okay. So here the vesicle forms. Here we have clathrin and AP2 coat. The whole vesicle forms and then decoating or the depolymerization of the coat happens. Then the whole structure here, the iron in the ligand, that is ferrotransferrin, and the ferrotransferrin receptor, the whole setup goes into the late endosome. In the late endosome, the low pH causes the release of iron, not the ligand. That is unique about this particular picture. So the iron gets released from the ligand, not the ligand itself, and Fe3 plus ions goes outside and gets reduced to Fe2, that is ferrous ion, by endosome reductase. Okay. What happens to this ligand and receptor? Well, this ligand and receptor without the presence of Fe3 plus ion is now called apotransferrin. The ligand itself is called apotransferrin. So this apotransferrin plus this transferrin receptor now gets recycled back to the cell surface. That means ligand doesn't detach from the receptor even at low pH. Okay. So the apotransferrin and receptor, when it reaches the surface, plasma membrane, then due to the neutral pH, that is pH 7, this apotransferrin releases from this transferrin receptor. The transferrin is glycoprotein in blood that is transports iron from liver, that is iron storage, and intestine, that is iron absorption. Here the transferrin receptors are present. So there is this late endosome, which is also known, as I told you earlier, there is curl compartment, CURL, that is compartment of uncoupling of receptor ligand. But in this particular case, the ligand and receptor does not get released even at the low pH. Okay, so few important points. What is the difference between ferrotransferrin and apotransferrin? Ferrotransferrin has iron ions or the ferric ions in the dimer. This one doesn't. Formation of AP2 clathrin coat, transferrin receptor, late endosome, metalloreductase reduces the Fe3 to Fe2 plus ions. Okay, and what else? The receptor and ligand both together gets released back to the cell surface. So I think we'll stop here, but for the exam purpose, I can tell you how to study. So for example, if you're studying for the exam, focus on the pictures. It is mostly memorization, and I know, because in this process I cannot give you some kind of question which where you have to think a lot because of time constraint. And if I give more time, obviously, since it is already an open book kind of exam, so students will get more and more time to basically just look at the book and write the answer. So there are several constraints, so I cannot give you any thought-provoking questions. I'll give you pretty simpler questions, and most of them will can be solved if you can memorize. But obviously, before memorizing, I would request you that please read the question carefully before answering. If it is a multiple choice question, please read the question carefully. To give you little more difficulty, I might change the sentencing of the question or maybe modify it a little bit so that unless you actually read and understand what the question says, it might not be the answer what you actually thought. Okay, so please go over the questions carefully. And for example, if you know the background, if you obviously that includes memorization, but if you know the background, if you know the concept, it should not be difficult for you to answer the questions. Okay, so for example, whatever we have studied so far, let's take a moment and see how much you know. So name two of the enzymes responsible for formation of M6P tag or addition of M6P tag to the proteins destined to go to lysosome. And where are those enzymes found? Those enzymes are found in cis Golgi. The first one is glucanac phosphotransferase, and the second one is phosphodiesterase. So for the soluble lysosomal proteins, where do you think the M6P receptor will be found? And what would be the coat protein? So since it's soluble lysosomal protein, that means it needs to go to the lysosome, that is internally. So the M6P receptors will be found on trans-Golgi network membrane, okay, TGN membrane, and the coat formation that will happen will be clathrine plus AP1 coat. Remember that, clathrine plus AP1 coat. But what about the secretory lysosomal proteins? That means the proteins which are already secreted outside the cell and are to be taken inside through the M6P receptor attachment. So for those case, we need to have the receptors on the plasma membrane, and the coat formation will happen with the help of clathrine and AP2 coat. Okay, important. What's the concept of eye cell disease? Cells lack one particular enzyme which inhibits the tagging of M6P receptor to the protein. What is that enzyme lacking? That is glucanac phosphotransferase. So these are the kind of questions you can question yourself while you are reading the lecture, or I mean listening to the lecture, or reading the material, or writing the notes. You can come up with questions and make a a list of questions, and also write the answers. Similarly, if you understand a topic, if you understand a picture, it should be very, very easy to be able to answer, because I won't go anything beyond what I have taught you in the class, or rather online. Okay, so all the best, and Wednesday we will go over another chapter. And for the exam, since it's happening on next Monday, so we'll have everything included till this Wednesday, whatever we'll learn. Everything will be included. And what else? Yeah. And also on Wednesday, I might give you some of the review if possible, and we'll see what we have learned so far. Okay. All the best. Bye-bye. Welcome back, students. So this week we'll start with chapter 17, but this is the slide where we actually stopped last lecture. And this slide is particularly important because we can see that unlike the M6P receptor or the LDH receptor, in case of transferrin cycle, the receptor and ligand doesn't get dissociated in the late endosome, irrespective of having the low pH. So that is the uniqueness of this transferrin cycle. Now, coming to the lecture 6, that is chapter 17, cell organization and movement. But before going into the details of this chapter, a few pointers about the exam. I have been getting some emails from students requesting me or asking me whether the exam is going to happen on next Monday. Yes, it will be on next Monday. Everything will run according to the syllabus unless it is mentioned otherwise. The exam will obviously be online, so I cannot give you short answer or very short answer or things like that I have already explained earlier. So what we can do is we can either go over the multiple choice or the fill in the blanks or crisscross, that is matching some or true false, something like that. Okay, I sincerely have not decided what are the questions and what will be the distribution of the questions, but do remember that the hour exam hour will be the regular class hour, that is 11:30 to 12:45, and the exam will be at the same time, and every single person, every single student from the class will have to take the exam at the same time, that is next Monday 13th at exact 11:30 a.m. and the exam will stop at 12:45 p.m. So The only exception will be the students who are registered with OSA. For them, there will be an extension of 1.5 hours. Other than that, all the students will have to take the exam on the same day at the same time. If you come across any issues, you'll definitely let me know or email me. But do remember that two of the major issues that was anticipated earlier by all the faculties during our discussion, which was the first is availability of electronic devices, and second is having a stable internet connection. Several suggestions have been suggested on announcement, and those suggestions were not compiled by me, but compiled by everyone and like given by everyone and compiled by the Associate Dean of NSM. So you need to take care of those things. Other than that, I don't anticipate any issues during the exam. Uh, but again, if you feel something is off or something is some answer is wrong or something like that, in that case, please let me know. Email me, and I'll see if it is true. I'll change it for everyone. And if it is not true, then obviously it won't be changed. And the exam will be fairly simple based on only the material. You don't have to think too much about the material, as long as obviously it includes a large part of memorization of the subject, because we cannot do otherwise at this point of time. As long as you know the material, it should be all right. But blank memorization may not help you in some of the questions. That is why I always say that read the questions carefully before answering. So read the question carefully before answering. You might see that the question says something else. So in that case, your answer might be changed. So you might need to think at least for 5 seconds or 10 seconds. That should be good enough. The other thing is the question timing. I haven't decided yet what will be the question time, whether it will be 1 minute per question. That will be too much actually. So uh, I cannot tell you at this point of time, but there will be an announcement and all the details will be given before this weekend. So wait for that, but then just prepare as you are preparing for the exam without even bothering about what will be the time frame and stuff like that. Because if it is enough time for you, it is enough time for pretty much everyone else except the OSA students. So don't worry about that. So anyway, having said that, I received a very good question uh, earlier from a student regarding the homework that I'm going to discuss. So after I discuss this chapter or today's lecture, I'll do a very brief kind of review for the exam and mention the important points and the points that you need to focus on. And most probably, I'm going to discuss that particular question, that interesting question from the student as well. I was very happy that the student actually went ahead and studied the material, also went over the lecture details, and came up with all the pointers. So it's very good, good job. Anyway, so chapter 17, the cell organization and movement. So the question is, why are we studying this chapter? That's the first question. So we are studying this chapter because this chapter involves with the cytoskeleton. Cytoskeleton means the skeleton inside the cell. Okay. So for example, if we study human system, so there are molecular systems, there are physiological systems, excuse me, there are muscle systems, and all the other individual organ systems. But do you think that body will exist without the bone structure? It won't. It will collapse, right? Uh, at least in case of human beings. Yes, there are some invertebrates who do not possess the bone structure or bony structure, but that is different. To maintain the integrity of the cell, these cytoskeleton elements are extremely important. Also, the interesting part is that these cytoskeletal elements actually can polymerize and depolymerize as needed. So it's a reversible polymerization. Also, the fact is that they are formed and in different ways. They basically they arrange in different ways and serve different purposes. So these are very interesting facts. Now we have three different components of the cytoskeleton. First is microfilament. The forming unit or subunit is called actin. So when several or actin or rather G-actin. So when several actin subunits come together and form a long filament, we call it a microfilament. Then we have microtubule, which is formed by alpha beta tubulin dimers. In this case, each dimer of alpha and beta tubulin comes together to form a long microtubule. The intermediate filaments, that is the several filaments coming together of in between sizes. Okay, and they serve different purposes. So for example, cytoskeletal regulation, if there is something wrong in particular cases, it can give rise to grave diseases as big as cardiomyopathy or metastatic cancer. So metastatic cancer means the cells in the tumors basically gets released in the circulation and move to another part of the body. So those cells get that locomotive feature by having irregular cytoskeleton regulation. Okay, so a single cell, in fact, can have all three, or most of the time do have all three filaments in its cytoskeleton, and each of the filament system has a distinct organization in the cell. So you can see that there are different colorings here. Here is red, green, and blue. So that means that each filament types can be labeled with specific fluorescent antibodies, which will give it a specific color, and we can actually track it under the microscope. Okay, so coming to the next slide, we have actin, which is a highly conserved and abundant eukaryotic cell in eukaryotic cell protein, or rather, it is an abundant eukaryotic cell protein. These assemble and form different structures which has different functions, right? The interesting part is it can reversibly assemble, and it's called F-actin filaments when it is assembled together, and when it is a single subunit, those are called G-actin. Okay, G-actin assembled to form F-actin. It's composed of two protofilaments, forms a helix, and blah blah. We'll just discuss this later on. So coming to the structural assembly and how they serve different purposes. So you can see that this pink line or the red line is the microfilament. Then this green line is the microtubule, and this blue line is the intermediate filaments. Now see the arrangement in a cell. This is the extracellular matrix. This is the base or the basin. Okay. So in this case, this is the basolateral domain, this is the apical domain. The microvilli are formed with the help of microfilaments. But the cell structure or the cytoskeleton integrity towards the wall or towards the periphery are being provided by the specific arrangement of microtubules, intermediate filaments, as well as the microfilaments. So these kind of arrangement basically helps in terms of nutrient import and export for the cell. But in other case, where the cell is basically motile, for example, WBC macrophage, here it is the epithelial cell lining of the intestine. In case of WBC macrophage, where the cell has to be motile and move to a direction, in that case, the cytoskeleton integrity is being provided on the periphery by the arrangement of microfilaments. These increased edges, also known as the leading edge or the filopodium, these are again being held by the specific arrangement of microfilaments. And in between, the specific arrangement of all three sorts of cytoskeletal elements basically gives it a proper structure. It also determines the cell polarity, helps in the cell division. Also, it involves, or the function is chemotaxis, phagocytosis. So we'll see what phagocytosis and chemotaxis means. So here is just a brief example. This is not part of the exam, but just to show you what is the difference in case of phagocytosis. So we have this circulating cell here. Okay, that is neutrophil in this case. The circulating neutrophil gets leaked from inside the blood capillary to outside the cell, and then whenever it comes across these microorganisms, they get engulfed by this neutrophil. The process is called phagocytosis. And once the phagocytosis happens, they get basically destroyed or degraded inside, and eventually the cell goes to apoptosis, that is cell death, automatic cell death, or self-decided cell death. I mean, we can have discussion over apoptosis as a topic for the entire semester. It's that big. But anyway, we are not going to discuss about apoptosis. And coming back to the chemotaxis, in this case, the neutrophil actually tumble upon, and then finally it gets released, but then it goes towards a direction where there is a chemical release. So it moves towards the chemical signal, also known as chemotaxis. So what basically prompts the cytoskeleton to arrange in specific ways? Those are basically the signals from the soluble factors, other cells, or from the extracellular matrix. So for example, in case of chemotaxis, we just saw that the chemical signal or the chemicals that's released in the surrounding basically prompt the cell to move towards that chemical, right? So in this case, that chemical becomes the soluble factor, okay? And it signals the cell, and there is a signal transduction pathway. And we have these plasma membrane with receptors. Most of the time, these are GPCRs, that is, G protein coupled receptors. The signal transduction pathways goes down inside the cell, and it basically instructs the cytoskeleton to arrange in certain way, which can involve either organization and movement of organelles, or gives a proper cell shape, or helps in movement and contraction and several other things. Okay. So that is why cytoskeleton is extremely important. Okay. Excuse me. Now we have the cytoskeleton, specifically the microfilament. So whenever we talk about microfilament, we talk about basically F-actin, because that is the polymerized form. The unpolymerized form, that is the single subunit, that is G-actin. Okay, this is the size, this is the size. I'm not going to ask you about this size for the G-actin. It is a bilobed structure, as we can see, and we can actually make 4 compartments, 1, 2, 3, and then 4. The interesting point is that it has a binding cleft for ATP or ADP. The other important point is that it also has a cation, that is magnesium ion. Okay, so when this subunit basically arranged in a specific fashion, then it is called F-actin, or basically it forms the microfilament. This one unit of F-actin is about 72 nanometer long, and it is formed by 2 of the filament coiled together, and each of the filament will have 14 subunits. Okay. This microfilament, this one unit of microfilament, always have polarity. Polarity means differentiation between the differentiation between one end from the other end. So whenever you see these binding clefts, these binding clefts are actually forming the negative end, and this round shaped non-binding cleft end forms the positive end, and we can actually see these kind of structures under the TEM or transmission electron microscope. So based on these informations, let's do this question answer. Effect of ATP, ADP and magnesium, potassium or sodium, any kind of cations. So what is the effect of those? Well, if it is ADP bound, it cannot form this F-actin. If it is ADP bound, then it is in a soluble state. That means a monomeric state. The other thing is, if it doesn't have magnesium, the polymerization rate will be, or the if the environment doesn't have enough cation concentration, that will affect the polymerization rate of this, or the assembly of this uh, single subunit to form these multi subunit structure that is called F-actin. So there are total of 6 different human actin genes, but surprising enough, when they were crystallized and the structure was seen, almost it was found that 93% of them are identical in terms of structure. Okay. Uh, there are other facts that we are not concerned with right now, or the other details, because I'm not going to ask you how many different types of actin genes are there. Okay. So now going to the dynamics of actin filaments. So actin filament polymerization, in vitro mechanism, kinetics, critical concentration, what these terms mean, and also regulation of actin filament polymerization. Okay. So let's start with the 3 phases of the actin polymerization, or basically the G-actin polymerization. So this is this experiment is actually done in vitro, that is outside the cell in a test tube. So what was seen is that the scientists provided isolated G-actin subunits. Over a period of time, they figure out that some of the units assembled together, specifically 3 ATP version G-actin monomers, form come close together to form a trimer, and that kind of serves as a nucleus, okay, on which other subunits keeps on getting added from both the ends. And here we will see that why this is needed later on, but then here we can see that the units are getting assembled from both ends. But then it was also observed, so this is the nucleation phase, and it was also observed that in the elongation phase, this particular end, that is the plus end, see the cleft here, that is the minus end. So this particular end, plus end, have more and more addition or faster addition compared to the negative end. And in the steady state, we can see that this whole structure is steady. We are also getting units disassembled from both ends at different rates. We'll see that later. And it basically gives it a stable structure. It suddenly doesn't get disintegrate from here or here. So it is basically maintaining a stable structure. So whatever units are coming in are also eventually going out. Okay. And also at the plus end, the addition of subunits is about 10 times more compared to the addition at the minus end. So what happens is that if we just plot this experimental details in a graph, we can say that this nucleation phase is basically a lag phase where we wait for this nucleus to form, the 3 subunits coming together, and that creates a little lag in time. And then once that happens, then there is rapid elongation from both the ends. And once this structure forms and it gets stable, then we call it a steady state. Okay. That means this particular unit is not going to get affected. Units will get assembled here, units will get disintegrate from here. Either it will stay the same or it will keep on increasing later on. Okay. So how can we get rid of this lag phase? For example, if we just provide the system with already made nucleus, the 3-unit assembly. In that case, once we add the nuclei, we will get rid of this lag phase. So we will directly go into the elongation phase. Once it acquires a specific length, then it will become steady state. Okay. So addition of short actin filament or nuclei or seeds can bypass the slower nucleation phase. Okay, and also we see that ATP is essential. So both addition of ATP as well as the already formed nuclei can actually increase the process or increase the rate of formation of F-actin. Now, if we see what is called CC, that is the critical concentration. In that case, we'll realize that CC is something or some particular concentration which is absolutely needed to start forming the filament. So let's say that there are 10 units that is absolutely needed to actually start forming the unit. Okay. So if there is, let's say, independent units, 9 units or 8 units, that is below CC concentration. So when we say total actin concentration, there is monomer and filaments together here. So when it is below CC, then the filament do not start forming. At CC, exactly when it reaches 10 subunit, for example, just hypothetically speaking, exactly at that point the filament will start forming. But the rate of polymerization and depolymerization is still going on. And once the filament lengths keep on increasing, and in that case, it will go into a steady state. In this case, you have to increase the monomer concentration till the point it reaches the CC, because at that point we'll reach the concentration which is needed to form the filament, and then it goes to a straight line. Do we not need more monomers to form the filament? We do. But then once it starts forming the filament, then assembly is happening at the same time disassembly is also happening. So the overall concentration of monomers and the microfilament stays the same pretty much. Okay. Before CC, filaments cannot assemble. After CC, filament starts assembling. And as we keep on adding monomers into the unit, then the filament length will increase as needed. Okay. So critical concentration for polymerization. Below CC cannot assemble, above CC filaments assemble, and G-actin addition is balanced by G-actin disassembly from filaments to yield a steady state mixture of both G-actin units and filament. The mass is equal to total actin concentration minus critical concentration. Okay. What is this? This is called actin treadmilling. So for next 3 pictures, it'll be very, very important. What is actin treadmilling? So for now, we know that there are several facts already established. The first one, we have G-actin subunits coming together to form F-actin long filament called microfilament, first of all. And then second is that the polarity decides the negative end will have this cleft structure, the positive end will have this a rounded structure. Okay. Also, we know that the assembly can happen from both the end, although the assembly at the plus end is almost 10 times more compared to the assembly at minus end. And the disassembly also happens from both the ends at almost similar rate. So if we see here, this is the rate of assembly at the plus end, which is about 12 micro per mole per second. In that case, and in this case, this is about 1.3. So if you multiply 1.3 into 10, that is about 13. So which is almost close to 12. So that is why we say almost 10 times more faster addition. Here the rate of disassembly 1.4 per second unit, 0.8 unit per second. So it's there. So that means that it cannot just keep on getting added on both the ends, because we, in that case, all the subunits will get depleted from the system. We need to have a suitable balance between the G-actin subunits and the F-actin filaments. So how do we determine the CC? CC is determined by dividing 1.4 divided by 12. That is about 0.12 micromole. That is the critical concentration. In this case, the critical concentration is 0.8 divided by 1.3, about 0.6 micromole. So where the critical concentration is more? This one or this one? Obviously this one. That means at the negative end, to increase or to get the subunits added at the negative end, we need to have more subunits available, and only then it can start polymerizing at the negative end. But in the plus end situation, even if we have less amount of less concentration of subunits, it can still polymerize. Obviously, that less concentration has to be more than critical concentration for sure. Okay, so that's what these critical concentrations tell you. So what is actin treadmilling? So it was just hypothesized that, let's say, these are the two subunits. This is obviously one subunit, but these are the two subunits. So we'll see why it is two subunits later on. But let's say from the plus end, forget about all the details here. Just assume that the plus end is only adding the subunits for now. In this case, we are getting the addition of ATP version of G-actin subunits added to the plus end. And then we know that the addition rate is much faster compared to the addition rate or dissociation rate here, or overall dissociation from here or from here. So once it adds, then it keeps on getting added. And in that case, what we see is that it is moving this unit. If we paint these units, we'll see that eventually it appears to be, it's not moving, but it appears to be moving towards the minus end. Why? Because we are having more and more subunits getting added at the plus end and less subunits getting added at the minus end, as well as less and less depolymerization. Okay, so overall polymerization overpowers the overall depolymerization, and it seems like if we start with one particular set of subunits, it will appear to go till the end until it basically depolymerizes. The other important factor is that once these subunits gets polymerized or added at the plus end, they are in the ATP version. In the middle, one phosphate is lost, so that is called ADP plus PI version. Okay, and once it reaches towards the end, at this particular point, or this area itself is called, if we divide about 1/3 portion, let's say approximately, in this later 1/3 portion, it is just ADP version. So what is getting added is ATP version of G-actin subunits. What is getting released is ADP version of G-actin subunits. So at steady state, ATP actin assembly on the plus end, which has a lower CC, is faster than actin ATP hydrolysis in the filament. That is overall actin ATP hydrolysis in the filament. Okay, so that is the concept of actin treadmilling. That two units appear to be moving from plus end to minus end over a period of time because the rate of polymerization at the plus end is faster compared to the overall ATP hydrolysis in the filament, and ATP version gets added and ADP version gets out after the hydrolysis of ATP. Okay, so that is what is all about actin treadmilling. Usually I used to give a short answer question on that, but I cannot give you that. But I may ask you what is the critical concentration at the plus end or the minus end, or what kind of version of actin is present at what stage, things like that. Okay. Coming to the next slide. So we were talking about how the rate of polymerization work at the plus end and the minus end. What is apparently the actin treadmilling. Now the question is, what happens or what are the other molecules that determines these association or the addition or the polymerization to form these F-actin microfilaments. So the topic is regulation of filament turnover by actin binding proteins. That means the proteins which actually bind to the actin subunits. Okay. So the first cycle, let's say this is the first cycle. Forget about this third one here. So it starts here where it is D, that is ADP version. T means ATP version. This is called profilin, cofilin, and thymosin beta 4. So these are the three proteins we will talk about. So the first cycle is called profilin cycle. In this case, we have the ADP version of isolated subunits where the profilin molecule gets added at which end? Minus end or the plus end? Remember the cleft side is minus end. So it gets added at the plus end, and then the ADP, basically that profilin removes the ADP and replaces it with ATP. Now it is ATP version of profilin associated. This goes and binds to the plus end. Okay. So this goes and binds to the plus end, and also it makes sure that since it is bound here in the plus end, that this cannot go and bind here at the minus end. Basically, it kind of regulates. So here it is mentioned profilin binding sterically blocks ATPG actin's assembly at plus end. It will be profilin binding sterically blocks ATPG actin assembly at minus end. It doesn't completely block it because we do see some addition from the negative end also, but it is much lower, and its sterical hindrance basically creates that lower rate of assembly. Okay, the second cycle, and once the unit gets added at the plus end, the profilin gets detached and goes into recycling mode. The second is the cofilin cycle. The cofilin is this particular yellow circle, this protein. What cofilin does is cofilin binds to two ADP G-actin monomers and induce a twist. So basically the cofilins gets added in between two ADP, because you remember this beginning is the ATPG actin, this is the ADP and PI version, this end is the ADP version now. So hydrolysis has happened here, so this is the actin ADP version. So here cofilin basically inserts itself between different subunits or two ADP subunits in multiple places and creates a twist. And that twisting basically cause it to detach a segment, like a chunk of this microfilament from this whole strand of F-actin filament. So now we can see, because of the twisting done by the cofilin addition, a chunk has been detached, and that makes sure, so then individual units gets dissociated, and obviously the cofilin goes for a recycle phase. What is the reason for doing this? Because remember, if we do not depolymerize the negative end, then we'll deplete the concentration of, or the reservoir of, single subunit G-actin. In that case, once that is depleted, we cannot have any more addition at the plus end. Okay. So the third one. So remember, initially we mentioned that the cytoskeleton structure is formed with the help of extracellular signaling. Whatever signal comes in from outside to inside of the cell, based on that, particular arrangement of cytoskeleton assembly happens. In this case, let's say that there is no signal for the moment, but we have a steady state reservoir of G-actin subunits and F-actin subunits. In that case, we do not need to create more of the F-actin filaments. So what needs to be done? That we need to sequester those freely available G-actin subunits. So once this stage appears, this ATP version of G-actin subunit, thymosin beta 4 is the one which buffers the ATP G-actin and sequesters the G-actin at higher concentration. And when the cell actually need it, they release the G-actin at lower concentration to polymerize. So that means thymosin beta 4, that is the cycle 3, it provides a buffered reservoir of ATP G-actin for polymerization. Okay. Then coming to the last slide for today's lecture, there are certain filament capping proteins which are essential in terms of capping the ends, either the plus end or the minus end. Now you might ask, what is the need of capping the plus end or minus end? Well, when we see the structural assembly of F-actin filaments in case of Filopodia or the growing end of a motile cell, we'll see that we need to curb the length of the filament as per the need. We cannot have continuous extension of a F-actin filaments, right? So in this case, the capping proteins comes in handy. So for example, the plus end capping protein, one of the protein is called CAPZ. CAPZ protein caps the plus end and inhibits any more assembly of subunits at the plus end. But the minus end is free. So the addition and polymerization and depolymerization is going at their own rate. So we can have a CC, that is critical concentration, for the negative end, but not for the plus end, because plus end is not doing anything. Similarly, for the negative end capping protein, the negative end is capped. So it's not doing anything, but the plus end, a regular process of polymerization and depolymerization is going on. So the critical concentration is being maintained here. Okay. So come back to the plus end capping protein, the CAPZ. The CAPZ is also regulated by a plasma membrane protein. It's a membrane protein called phospholipid 4,5 biphosphate Pi45P2, also known as phospholipid phosphatidylinositol. So I don't want you to remember the whole name. If you can, that is wonderful. But 4,5 Pi45P2 should be good enough. Okay, and also it is a plasma membrane. It's a regulatory protein. So regulatory protein, remember, through the regulatory secretion, when it is needed, only then it gets secreted. And also some other regulatory proteins. The second Positive end capping protein is called gelsolin. It inserts and severs the actin filament at the plus end, which is activated. The whole process is activated by the presence and, in fact, increase in cation concentration. There is the calcium ion concentration. So once the calcium ion concentration is high in the cell, in that case, the plus end will get cleaved by insertion of gelsolins. Okay, remember insertion of gelsolin and cleaving, something that sounds similar, that we have already seen in case of cofilin, because cofilin attaches itself in the minus end and basically severs chunks from the minus end. In this case, gelsolin binds at the plus end and severs chunks from the plus end. In case of this, the negative end capping protein, we have tropomodulin. What tropomodulin does is tropomodulin binds with the tropomyosin and binds, this assembly binds at the minus end, and it greatly stabilizes the filament because not much of disassembly is happening here, but the assembly at very fast rate is going on in the plus end. That means this area will stop doing whatever it was doing at the minus end, but this area will keep on increasing in length. So let's say we have a cell surface here like a arch, and we need to basically make a indent on the arch surface. So we can have the assembly of this microfilament at that point, and basically that will push the boundary by keeping on increasing the plus end. So these are some of the background details about the actin filaments, how the assembly happens, and also the capping proteins responsible. We will talk about the actin nucleation and the long filament assembly versus the side filament assembly in the next lecture. So we'll keep this lecture short and one single unit, and I will also try to make a brief video of review for the exam 3, uh, and we'll go over from there. Okay, so all the best for the exam. Wait for the announcement, but in the meantime, keep on studying as you are. If you come across any issues, let me know, and do take help of the workshops, attend the workshops, also go over the workshop materials or the recordings as many times as you want, and ask any questions that you want from TAs. I mean, you can just email TAs, undergraduate TAs, your workshop TAs, or even the graduate TA, doesn't matter. And even if you are not satisfied with the answer or you need to know more, then obviously you can email me as well. Okay. So all the best for today. And we will meet online next Monday for the exam. Again, Monday, the exam will start exactly at 11:30 online. You will have a link on e-learning uh, in the second part of the semester folder in my folder, and I'll mention about that in the announcement before by the weekend, and you can click on there and the exam will start. You most probably will not have the backtracking option, so you are going to see only one question at a time, and once you click on the right answer, only then you can move on to the second question. Also, you will not have the option to move back at the end of the exam to see all the exams and review them, because it is online test and it's not done usually. The other thing is you cannot change the answer. Like once you go back, it's the same thing basically. So once you done one question, once you answer one question and go to the next question, you cannot come back to the previous question and change the answer. Okay, so it is one time submission only. And once the time ends, except OSA students, once the time ends, In that case, that is 12:45, 1 hour 15 minutes. Once the time ends, your exam will be done. Whether you are done or not, the exam will kick you out. The e-learning basically will kick you out. And whatever you have done will automatically get submitted for grading. Okay. And you can have your grades, I mean, it will show up uh, soon, but most probably I'm thinking of keeping it off till this weekend or probably the mid of the week, because if there are some questions or some doubts, we can clarify them, and then as needed, then we can publish the whole grade to the whole class. Okay, so all the best for the exam. Keep on studying and good luck. Bye bye.